Welcome to the cell topic and its discussion. In the previous session, we have already seen or referred to some of the subcellular organelles. So today we will be discussing some of the subcellular organelles. So let me begin with endoplasmic reticulum. So it is one of the most important subcellular organelle which are 50 to 100 nanometer in diameter. These are the continuous membrane system which is of two types, the small endoplasmic reticulum and rough endoplasmic reticulum, which contains tubules and flattened sacs. So these endoplasmic reticulum are helping in calcium storage, protein synthesis and lipid metabolism. Next subcellular organelle is F0 F1 particles. So these are present in mitochondria and they are 10 nanometer in diameter and 20 nanometer in height. They are also called as oxysomes. And this, as we all know that mitochondria is having two membranes, outer membrane and inner membrane. The inner membrane have some folds known as cristae. So the F0, F1 particles, or I can say oxysomes, are present on the cristae or inside the cristae or on the folds of the cristae. So these F0 F1 particles are helping in ATP production and oxidation. So moving on to the next subcellular organelle, flagella. So the diameter of flagella is 0.01 to 0.02 μm and length up to 10 μm. It's an hair-like structure or whip-like appearance. At the time of basal body, we have seen basal body is present at the base and flagella or cilia is arising upon that. So the elements of the flagella is basal body, hook and filament, which helps in cell movement and chemotaxis. Next is Golgi complex, or I can say Golgi apparatus, or it is known as Golgi apparatus 2. The diameter is given of Golgi apparatus is 0.5 to 2 μm, and these are the stacks of flattened membrane cisternae. So like this they are arranged one above the other, which are helping in transporting, modifying and packaging of proteins and lipids into vesicles for delivery to target destinations. So these proteins and lipids, they are packed into the vesicles by Golgi complex and they are delivered to target destinations. Upon reaching there, they will get released and start showing their functionings. Next is histone proteins. They are present inside the nucleus. The measurement givens are 11 to 15 kDa. These are present as or observed as histone octamers, set of 8 proteins which are of 4 types, H2A, H2B, H3 and H4. So these histone proteins, they are wrapped by the DNA. So they are associated with the DNA and providing structural support to a chromosome. So moving on to the next subcellular organelles or other subcellular organelles such as intermediate filament. So we have already seen in the section of cytoskeleton, there are three types of cytoskeleton: intermediate filament, microfilament and microtubules. So these are ranged between microfilament and microtubules, that is 8 to 14 nanometer. So these are the filamentous structure which are medium sized protein filaments. They are submerged or present in cytoplasm. They provide structure and support, and they can withstand a mechanical stress during wear and tear of the organism or cell. Next one is lysosomes. So these are one of the smallest subcellular organelle of a cell, which are 200 to 600 nanometer in size. They are known as suicidal bags or digestive bags because they break down cellular waste and debris by engulfing it, or they are having digestive enzymes and cytokines too. They are showing the antimicrobial properties. So what they will do is lysosomes, upon upon identifying a debris or waste material which are present into the cytoplasm or around the surroundings, they will reach to that position, and after reaching it, what they will do is they will start secreting hydrolytic enzymes and they will start engulfing it. Upon engulfing it, it will take them into them. This waste material will come inside and they will get broken down into small pieces. So by this enzyme or by this process known as phagocytosis, they will remove the debris inside of the cell. Next is microfilament, which is a type of cytoskeleton. So it is 5 to 7 nanometer in diameter. So they are known as actin filaments. So in the section of actin filaments, I have told you that we will read in microfilament. So they are principally of actin protein subunits. They are small protein subunits. What they will do is they will polymerize to form elongated actin filament. They are used for structure and support in the cell, as their name indicates, cytoskeleton, and they are separating cell during cell divisions. One cell will get separated into two cells during cell divisions with the help of microfilament, and they allow motility and changes in cell shape. Next, one of the cytoskeleton is microtubules. Already we have completed two cytoskeleton, and this is last one, that is microtubules. They are 25 nanometer in diameter, or I can say polymers of tubulin. These are polymers of actin subunits. These are polymers of tubulin such as alpha tubulin and beta tubulin subunits, and they will get assembled into linear protofilaments like that of actin filaments. So they are helping in mitosis, cell motility, intracellular transport, and maintenance of cell shape. As name is their cytoskeleton, so all the cytoskeletons are helping in maintenance of cell shape. Next is mitochondria, which is also one of the most important subcellular organelle of cell. The size is 0.5 to 1 μm. So this name is given as powerhouse of the cell to the mitochondria, as it is producing ATP in much amount, and it is present in both animal and plant cells and contains ribosomes. It is a primitive subcellular organelle which can be found in mitochondria. It is a primitive character of mitochondria that ribosomes are present. It helps in ATP production, maintenance of cell growth, and cellular differentiation. So by this we have completed some of the other subcellular organelles. So we will be looking after of the remaining subcellular organelles in our next section. Thank you.